Hello, I'm Dr. Mark Darcy, and in today's lecture, I'm going to be discussing epigenetics. Now, before I start, for anyone that's watching this particular lecture on YouTube rather than listening to it on a podcast app, you're probably wondering about this new look. The reason for this, the reason for the beanie, and the reason for the beard is basically COVID-19. So we're in the middle of the COVID epidemic at the moment, pandemic, I should say. And my hair was getting a little bit long before the pandemic even began. I was due a haircut. And then when the pandemic did begin and all the hairdressers closed, obviously I could not get a haircut. It was getting a bit messy, so I bought some clippers. You can see where this is going. And the wife, she basically went over my head with those clippers the other day. She made a little bit of a mistake, so I had to go over it again to correct the mistake with a shorter setting. Long story short, under this hat right now, I kind of look a little bit like a white supremacist, which isn't a look that I would like to go for. So I purchased a beanie. I'm covering the head until the hair grows back. So rather than looking like a white supremacist, I think I now look kind of like a hipster, especially as I've got this little ginger beard that started to grow over the last couple of weeks too. So that's why I look like this. It shouldn't take anything away from the quality of the lecture, hopefully, which we should probably get on with. So epigenetics, what is epigenetics? Well, there is the genome. The genome is basically the sequence of nucleic acids in our DNA, the sequence of A's, C's, T's, and G's, adenine, thymine, guanine, and cytosine. That is our genome, the sequence of bases. A group of bases together accounts for a gene. Genes, when they are active, when they are switched on, create something called messenger RNA. RNA. The RNA, unlike DNA, is single-stranded. This RNA then goes to a structure in the cytoplasm of the cell, the liquid of the cell called a ribosome. This messenger RNA is read by the ribosome and a protein is made. So for example, there might be a gene for creating insulin. So a sequence of bases that codes for making insulin. When that gene is switched on, then the double-stranded DNA will open. A strand will be read by an enzyme called RNA polymerase, and a single strand of messenger RNA will be produced, a message that carries the instructions from that insulin gene out to the cytoplasm, where it is read by a ribosome, and the ribosome will make the insulin protein. So that's basically how it works. It's known as the central dogma of molecular biology. So basically, DNA makes RNA, RNA makes protein. Now that's pretty straightforward. We have about 25,000 genes coding for many, many different proteins, and that is a genome. Now, the epigenome is a little different. The epigenome is basically which genes are switched on or off at any one time and to what degree. So epigenomes vary quite a bit. They vary within our body. So for example, a skin cell has got the same number of genes as a neuron in your brain, about 25,000 genes. But different genes need to be switched on and off and to different levels in those two types of cell, because those cells do different things, they have different functions. So for example, a neuron will have genes switched on that allow that neuron to make neurotransmitters such as serotonin or dopamine, whereas a skin cell will not have those genes switched on. Instead, it will have the sorts of genes switched on that allow it to grow and divide quite rapidly, because that's what skin cells need to do. Now, this can go wrong. When you're young, pretty much all the genes are switched on and off to the right degree for the type of tissue that they are. As you get older, however, there can be a change in the epigenome, in the patterns of activity. Epigenetics is basically the study of the patterns of activity of the genes within your cells, how genes are switched on and off, and to what degree. And that can change as you get older, making cells misbehave, but I will get to that later. Okay, so now let me just show out this desktop and get started. So, okay, so here we go. What we're looking at here is known as a karyotype. A karyotype is basically it's a picture of all of an individual's chromosomes at one time. So we have 46 chromosomes, 23 pairs. So you can see we have 2 chromosome 1s, 2 2s, 3, 4, 5, etc., etc. And then we have 2 sex chromosomes. So these chromosomes are known as the autosomes, whereas the X and Y, this is a male, XY, a female would be XX, are known as the sex chromosomes. So we have a pair of chromosomes, 23 pairs of chromosomes. One set are inherited from our father and one from our mother. So in this lecture, I'm going to talk about chromosome structure first, very briefly, and then I'm going to delve into epigenetics. So chromosomes. So first of all, what length of DNA do you think there are in eukaryotic cells? Our cells are eukaryotic, by the way. Bacteria are prokaryotic. Any guesses? You might be surprised. We have about 2 meters of DNA in every single one of our cells. 2 meters of DNA. And that's a lot of DNA for a very small cell. So to manage this, the DNA needs to be wrapped up with proteins into a structure known as chromatin. So chromatin is a complex of DNA and proteins. The DNA is wrapped around the protein so that it can be compacted into a very small space. So humans, as I said earlier, have 46 chromosomes. Different animals have different numbers of chromosomes. By the way, we don't have the most. There are plants that have far more chromosomes than humans do. So it's not the number of chromosomes that's important. It's more the genes you have and their level of activity. So chromatin is basically a complex of proteins and DNA. It condenses prior to mitosis. Mitosis is the process by which a cell divides to reproduce, to make daughter cells. Now, for most of a cell's life, the chromosomes are kind of more of a relaxed form. There's like a spider web of chromatin within the nucleus of cells. It's just before division that it all condenses tightly, very, very tightly, and then you can see the chromatin as chromosomes. This compaction is needed to allow the cells, the chromosomes to be easily separated following replication of the cell. So chromatin, what is it made of? So what we're looking at here is a picture of the nucleus of the cell. You can see some kind of light color and some dark color. The light color is basically the less condensed chromatin. It's lighter because it's less dense. And the darker material is more dense, more condensed. I'll get into that shortly. So this is basically chromatin spread throughout the nucleus of the cell. All the different chromosomes basically unraveled slightly and wrapped around each other. Chromatin consists of about 30% DNA and then 60% histone proteins. The histone proteins I'm going to get to in the next slide or two. Histone proteins basically are like balls of protein, and the DNA is sort of wrapped around these balls so that it can be compacted. So DNA is about 30% of chromatin, histone proteins about 60%, and the rest is RNA, about 10%. So this is arranged together into fundamental units known as nucleosomes. The proteins and the DNA together are known as nucleosomes. This condenses into bar-like bodies called chromosomes when the cell starts to divide. Nucleosomes, complex of DNA wrapped around positively charged proteins called histones. So I should say that DNA is negatively charged. Histone proteins are positively charged. That's what holds them together. So this is the DNA. Sorry, this is the ribosome. This is a ribosome. Ribosome, what am I talking about? I'm getting ahead of myself. So this is a histone protein. It's positively charged, and wrapped around it is the DNA, which is negatively charged. And because the DNA is negative and the histone proteins are positively charged, they hold together. That's what holds them together. The strength that the histone proteins of DNA hold together is determined by how positive the histone proteins are and how negative the DNA is, which can be modified. I'll get to that later. So histones form an optimal core particle. So there are basically 8 histone proteins in that ball with DNA wrapped around it. DNA wraps around the core particle slightly less than 2 times. Nucleosomes are the unit particles of chromatin. So you have a string of DNA, and nucleosomes, which form a helix about 30 nanometers wide. Then there is further looping and coiling and twisting, which condenses everything tighter and tighter. So this is what the core particle, the nucleosome, looks like. So we've got it from the front and from the side. So you can kind of see this set of proteins in the middle. These are the histone proteins, and wrapped around is the DNA. And you can see here again from the side. Nucleosomes, histone subunits. So if you actually do a drawing of this, you can see that we basically have 8 particles. So we've got 2H2A, that's a kind of histone protein, 2H2B, 2H3, 2H4. So 8 particles in a little ball. These are the histone proteins with the DNA wrapped around and then wrapping around the next nucleosome set of histone proteins, and the next and the next and the next. So this basically is the size, about 10 nanometers in diameter. You might wonder where H1 is, by the way, because we've got H2A, B, 3, and 4. H1 is sort of in between here, not shown in the picture. Okay, so this is how we order the chromatin. So we start off with the DNA, about 2 nanometers diameter. This wraps around the histone proteins to create nucleosomes, which is 11 nanometers. This wraps around itself again at 30 nanometers, and again 300 nanometers, again 700 nanometers, and finally once more to make 1400 nanometer structure known as chromosomes. This is how you get 2 meters of DNA into 1 nucleus. And this is a scanning electron micrograph. This is just before cell division. So what we have here, what we call homologous pairs of chromosomes. So these are chromatids. So if you take the chromosome 1 from your father, the chromosome 1 from your mother, just before cellular division, both of those replicate. So this could be, say, for example, your mother's chromosome 1 that you inherited. Just before cell division, it divides into chromatids. So we've got 1 chromatid here, 1 chromatid here. They get pulled apart so that they can be separated into daughter cells during mitosis. That's for another lecture, I think. Okay, so human chromosomes. They are diploid. We have a diploid number of chromosomes. That means 2 sets. In all cells except the gametes. So in all of our normal cells, we have 2 sets of chromosomes, 1 from our mother, 1 from our father. The gametes are an exception. The gametes are the eggs and the sperm. When we make eggs or sperm, we have the number. That way, if you go down to 23 chromosomes in an egg, 23 in a sperm, when they fuse together, you go back to 46. So there's 1 pair of sex chromosomes determining the sex for female XX, for a male XY, and 22 pairs of autosomes, the chromosomes 1, 2, 3, 4, 5. They are homologous. They are the same, more or less. So there's pairs of homologous chromosomes, 1 pair from the mother, 1 from the father. I should say that they're homologous, meaning that 2 chromosome 1s, the 1 from your mother and the 1 from your father, contain all the same genes, but they may have different versions of those genes. So perhaps you have an eye color gene that you inherit from your mother on 1 chromosome, and therefore from your father, you also have an eye color gene on the homologous chromosome. But there could be different alleles of the genes. One could be a blue eye color gene, one could be a brown eye color genes. So you have the same genes on your chromosomes, but you can have different alleles, different versions of those genes. For more about that, you might want to listen to my Mendelian genetics lecture. I go into more detail about genes and dominance and recessiveness and all that kind of thing then. Okay. The telomeres. The end of a chromosome has a kind of pad known as a telomere. You can see that in this picture here, where you basically, it's been stained so that it fluoresces the end of each chromosome. The telomeres are short repeating sequences enriched in the G base, guanine. Between about 100 and 1,000 copies of the following nucleotide sequence, TTAGGG, is at the end of every single chromosome. It acts like a pad, so think about a shoelace. Shoelaces have the bit of plastic at the end that protects the shoelace from fraying, from becoming damaged. And the telomeres at the end of our chromosomes also protect our chromosomes from damage. Now, these telomeres wear out over time, As we age, they get shorter and shorter. Eventually, they wear out completely or close to completely. And when that happens, our DNA gets damaged. That's part of the reason why we age. So variation in chromatin condensation. So there are two types of chromatin in a nucleus. There's heterochromatin and euchromatin. Heterochromatin is basically it's condensed chromatin that stays very condensed throughout the cell cycle. It's the darker material I showed you in the previous image. Basically, the condensed chromatin, the heterochromatin, it's not active. The genes in the heterochromatin are switched off. They are off because it's too condensed to be active. Then there is the euchromatin, the true chromatin. It condenses only prior to cell division. Transcription only occurs from euchromatin. Transcription is when a gene is switched on and you make messenger RNA. That only occurs in euchromatin, which is not highly condensed. So here you can see E for euchromatin, H for heterochromatin. The heterochromatin looks darker. It also looks like there's less of it. There's not. There's more of it, but it's condensed. The euchromatin is the lighter material that consists of the genes which are switched on. So let's now get into epigenetics more itself. Epigenetics is how you switch genes on or off. And as I said previously, we have about 25,000 genes. We have about 25,000 genes in our cells, and different genes are switched on in different types of cell. So some genes need to be switched off, some need to be switched on, some need to be switched off a little bit or on a little bit more. And we do that through methylation, chromatin remodeling, histone modification, and RNA interference. I'm going to go into these methods in more detail now. So first of all, DNA methylation. This is the method of switching genes on and off that we know about the most. So what we're looking at here is cytosine. So you can see cytosine. Cytosine is one of the bases in DNA. We have A, T, C, and G. Cytosine can be methylated. So you can see here in the second image, we have 5-methylcytosine. A methyl group, CH3, has been placed on cytosine. This is now methylated cytosine. Now, when you methylate cytosine, you sort of make it harder for that gene to be expressed. So when you put methylation when you methylate DNA, you make it more negatively charged. And as I said previously, we have positively charged histone proteins with negatively charged DNA wrapped around it. If you put methyl groups on the cytosines within a region of your DNA, you increase the negative charge, and that holds the DNA tighter to the histone proteins, making it harder for that gene to switch on. So for example, here, we can see we've got different alleles of genes. I'm not going to go into this image in too much detail, but I'll go through the basics. So here are some C's and G's. C binds with G. The P is a phosphate. So these are known as CpG islands, regions of DNA where there are lots of C's, lots of cytosines. Now, this gene is active, indicated by the arrow. Now, if you place methyl on these CpG islands, then you hold the DNA tighter to the histone proteins and the gene cannot be expressed. It is switched off. There are other mechanisms involved in this switching on and off. We have certain proteins known as activators, which can help a gene to become active, to switch on, can help unravel the DNA. And we have repressors, which help to switch genes off. These all interact with this methylation to determine how active a gene is, whether it's switched on, whether it's switched off. A gene can be switched on, for example, and therefore inhibit some repressor gene, a gene that makes a repressor protein. Or it can be switched off, and therefore that repressor is no longer deactivated. So you can basically switch on a gene, activate a repressor, or activate a activator. You can switch off a gene, etc., etc., etc. So there's all sorts of different connotations whereby you can switch on activators or switch them off, switch on repressors or switch them off, and it all boils down, at least partially, to how methylated particular genes are. Okay, so methylation maintenance. Now, DNA replicates itself when cells divide. Now, you don't want a cell to replicate its DNA, create a daughter cell that has different methylation patterns to the parent cell. You don't want a daughter cell that should become a skin cell to become a neuron, for example. So the way to keep a cell behaving the way it should when its DNA is replicated is to maintain the methylation patterns, so that the methylation patterns are closer to the parent cell, the parent DNA, than to the DNA in some other type of cell. This is how we do it. So, at least one of the ways we do it. So here we've got some double-stranded DNA with A's, C's, T's, and G's. This is normal unmethylated DNA. Now, methylation, methyl groups are added to DNA using various enzymes, one of which is known as DNA methyl transferase, DNMT. So now we have certain genes that have been silenced, that have been switched off within the DNA by having methyl groups added by the DNMT. Now, imagine this DNA now replicates. To replicate, it unzips, it opens up, and each of the single strands is read by DNA polymerase, and a new double strand is made from each single strand. So you can see that here we've got one double-stranded piece of DNA, which is now becoming two double strands of DNA. The DNA is being replicated prior to cell division so that you get the same number of chromosomes in each daughter cell. So how do we get the methyl on? Well, DNMT will come along and various other enzymes. And what we will see is they will look at the DNA as it's dividing. And to start with, the daughter strand will have no methyl on. So you've got one parent strand with methyl, daughter strand, the new strand, no methyl yet. Same here. Now, DNMT, what it does is it reads the parent strand. It looks to see if a strand is methylated, and then it places methyl on the opposite side, as you can see here. So we now have placed methyl groups on the DNA. So the daughter strand has now got the same methylation pattern as the parent strand. So that's fully methylated, and DNMT does that. The thing is, we don't always want to maintain methylation patterns. So, for example, when you make eggs and sperm, you kind of need to reset to go back to almost to factory settings. Because when an egg and a sperm fuse together and make a zygote, it needs to be reset. All the genes need to be reset in that zygote so that as the cells divide, you can make skin and muscle and brain and bone and all the other types of tissue. So you need to reset the methylation pattern. So you need to get rid of methyl in some cases. And to do that, you use enzymes such as 5-AZA. If 5-AZA is present, there's a number of other enzymes involved as well, then it blocks the methylation of daughter strands. So this DNA replicates, and now you've got daughter strands that are only half methylated, they are hemimethylated. And when they replicate, you'll have even less methylation. After a few replications, methylation has been removed. You have reactivated, deactivated genes, and they can be expressed. So you have a bunch of enzymes that can place methyl onto DNA, and you have other enzymes that can remove it, sort of reset the DNA expression patterns. Hope that's clear. Which leads me to imprinting. There are certain genes you will get from your mother, certain other versions of the genes you will get from your father. So you will get one set of genes from your mother, one from your father. They have the same genes. They might be different alleles, but they're the same genes. But when a zygote forms, so when an egg and a sperm fuse and you get a zygote, everything isn't completely reset. There are certain genes that seem to remember that they came from the father, and other genes that remember coming from the mother. Certain genes, certain alleles, certain genes. Now, this memory of methylation is known as imprinting. So we have here an embryo which develops and creates somatic cell lineages. Somatic cells are just the cells of your body, heart, liver, brain, etc. But there's also germline lineage, the eggs and the sperm. Now, these eggs and the sperm that are produced, they can maintain the methylation patterns, some of them can, that were taken from the parent, different in males, different in females, so that when eggs and sperm fuse together, you can end up with a new zygote that has got a certain specific methylation pattern. This is known as imprinting. Not going to go into imprinting in a lot of detail right now, but let's just say that it is quite important in biology. DNA methylation, X-inactivation. Now, I mentioned previously that males have an X and a Y chromosome, as well as many other chromosomes. Females have two X's. Now, the thing is, both of those X's cannot be fully active in the same cell. You would end up with too high a dose of expression of certain genes. So when a female is formed and she has two X chromosomes in her cells, one from her mother, one from her father, one of those X chromosomes in each cell needs to be switched off, at least mostly switched off. Only about 15% of one of the X chromosomes will be active. The rest of that X chromosome will be fully active, more or less. So we need to deactivate an X chromosome in each cell, and it's random. In different cells within a female, there will be maybe the father's X chromosome active. In other cells, it will be the mother's X chromosome that is active. So this inactivation involves DNA methylation. You hypermethylate one of the X chromosomes, and it shrinks and shrinks and shrinks and hypercondenses and becomes switched off. It's random in placental mammals. Which of the two X chromosomes is switched off forms heterochromatin. It's known as a bar body. Looks like this. The arrow indicates an inactivated X chromosome known as a bar body. And this has some very strong implications. We all have two chromosome 1s, two chromosome 2, 3, 4, 5, 6, 7, etc., etc. But if you're male, you only have one X chromosome and a Y chromosome. You're female, you have two X chromosomes. Now, this can lead to problems. X chromosomes have certain genes present on them. For example, in relating to color vision or relating to blood clotting. Now, if you're a female and you have a defective gene on the X chromosome, that might not be a problem because you have another X chromosome. In some of your cells, the defective X chromosome will be active, but in other cells, the good X chromosome will be active. So you may be asymptomatic, no symptoms of that particular disease. If you're male, however, you only have one X chromosome, and if that X chromosome carries some sort of defective gene, then that is going to be expressed in all of your cells, and therefore you're more likely to have symptoms. That's why men are more likely to be colorblind than women, because certain genes involved in color vision are present on the X chromosome. So if you have a faulty version, it's expressed throughout your body if you're male. Whereas if you're female, you'd need a faulty gene on both X chromosomes for it to be expressed. It's the same with hemophilia. So if you have a problem with blood clotting, you have hemophilia. And one of the clotting factors, the gene for it is located on the X chromosome. So if you have a faulty one and you're male, you're more likely you're going to have hemophilia. Whereas if you're female and you've just got one faulty gene on one X chromosome, well, the good gene on the other X chromosome will compensate. A good example of X inactivation and the effects of it can be seen in tortoiseshell cats, which are all female. Their color gene for cat fur is located on the X chromosome. So let's take this female who has two X chromosomes. Males only have the one, remember. Now, if this female inherited one X chromosome with the orange color gene for fur and another from the other parent with a black color, then in some of her cells, the black will be inactivated and the orange activated, but in other cells, it will be the other way around, which accounts for the patchy color of fur on a tortoiseshell cat. So histone modifications. I've talked about methylation, but methylation is only one way that we can mess around with the activity of genes. There is also acetylation, ubiquination, summylation, and phosphorylation. I'm really only going to talk about acetylation now. I've mentioned methylation. I'm going to talk about acetylation too. Acetylation is something that occurs on the histone proteins themselves. So here we have the histone proteins in the middle and the DNA wrapped around. The amount of acetyl groups on the histone proteins can affect the expression of a gene. So addition of acetyl groups to histones via the lysine residue. So lysine is an amino acid. Proteins are made of amino acids. So histone proteins have lysines in them, a type of amino acid. Now you can place acetyl group onto a lysine or you can remove it. So if you acetylate a histone protein, it has the opposite effect of methylating the DNA. So when you place acetyl onto histone proteins, you make the histone proteins less positively charged. And therefore, the DNA wrapped around the histone proteins becomes looser. So you can help switch on a gene by placing acetyl groups onto the histone proteins. If you remove them, however, you make the histone proteins more positively charged, and the DNA and the histone proteins will become tighter. And as a result of that, the gene will be deactivated. So histones are positively charged, so bind DNA negatively charged tightly. Acetylation of lysine and arginine residues neutralizes this positive charge, making everything a bit looser, helping to switch the gene on. So here we have lysine with acetyl placed on it. So we look here at some histone proteins with no acetyl. Here are some histone proteins with acetyls placed on them. It causes the DNA to loosen up and genes to be switched on. You can also place methyl groups on histone proteins to again mess around with the positive and negative charge and to help switch genes on or off. I'm not going to dwell on this slide. This just shows you that there are a lot of histone modification recognition proteins, proteins which can bind and change the charge of histone proteins and therefore help switch genes on or off. Now, there are interactions between different epigenetic markers. If you start switching a gene off, then this attracts other enzymes which help to continue switching the gene off. So, for example, if we look here in the slides, we can see we have an acetyl group here on some histone proteins. And here we have some methyl groups on DNA. So this gene is partially switched off, begun to be switched off. Now, there is a protein, MECP2, an enzyme. This protein binds to methylated DNA. And when it binds to methylated DNA, it attracts another protein known as HDAC. HDAC stands for histone deacetylase. It deacetylates. So the MECP2 bound to the methylated DNA attracts HDAC. And when HDAC binds to the region of DNA, it then removes acetyl groups, basically switching the gene off even more. So there's a kind of cumulative effect. Methylation promotes deacetylation, helping to switch a gene off further. DNA methylation is linked with aging and various other diseases. So I'm not going to talk about these diseases, but there's a number of diseases. So there's imprinting disorders such as Beckwith-Wiedemann syndrome, Prader-Willi syndrome, transient neonatal diabetes. All are controlled by methylation. So methylation patterns can be changed. These three are imprinting disorders. We have repeat instability diseases such as Fragile X syndrome and various muscular dystrophies. And we have defects of methylation machinery, which can cause diseases such as systemic lupus erythematosus, an autoimmune disease, and immunodeficiency diseases as well. So there's lots of diseases that can be caused by failures in gene regulation. There's also diseases caused by histone modifications. So Coffin-Lowry syndrome and Rubinstein-Taybi syndrome being examples. being examples of this. So all caused by various problems with histone modifications, causing a dysregulation of gene activity. Now, I'll briefly talk about microRNAs. So eukaryotic cells, and we consist of eukaryotic cells, humans. Animals and plants are all eukaryotic, code for many short RNA molecules known as microRNAs, so little pieces of RNA. So we make the messenger RNA, which goes to proteins, so it goes to ribosomes to make proteins, but we also have other RNAs that are coded for, known as microRNAs, about 22 bases in length. These microRNAs can regulate gene expression by base pairing with complementary sequences in target mRNAs. Let's summarize how this works. So you make your messenger RNA. So a gene has been switched on and it makes messenger RNA. This messenger RNA should go to a ribosome, get read, and then a protein is made. However, you can regulate the activity of this messenger RNA. So messenger RNA is made, but in another part of the genome, microRNAs are made, which match partially the sequence of the mRNA. And these microRNAs will bind to parts of the messenger RNA, and it will then be degraded by various enzymes. This double-stranded RNA will be degraded by various enzymes. I won't go into these enzymes right now, but there's plenty of them in our cells. So if the messenger RNA is degraded before it can get to the ribosome, the protein can't be made. So you can regulate the activity of a gene by making these small interfering pieces of RNA, which are complementary to messenger RNA, and they bind to it before it gets a chance to go to the ribosome and to make protein. So, aging and cancer. Both of these diseases are partially caused by misregulation of gene expression. So when we are young, our cells more or less behave the way they should. There are the correct methylation patterns in each cell type, more or less. So here is a normal cell. Many of its genes are methylated, hypermethylation. The genes that should be off, which is many of the genes, are switched off. Now, what can happen is over time, due to chemicals in our environment, due to respiration, due to living, due to radiation, gradually over time, the methylation patterns in a particular type of cell can change, and you can end up with hypomethylation. So basically, you end up with certain genes being switched on. They're not methylated anymore, but should be off. Now, imagine what that can do. So imagine you have a brain cell. Brain cells don't divide very often. But then there's a change in methylation sequence within some of the genes of that cell. And some of the genes which should only be on in skin cells get switched on in the neuron. Now, skin cells grow and divide rapidly. So now you could end up with a neuron that grows and divides rapidly. You then have a cancer cell. And this can happen throughout the body. So when cells lose the proper methylation pattern, genes can be switched on that should be off. Cells can start misbehaving and can become cancerous. But it's not just cancer, it's aging as well. As we age, there's more, basically the longer we live, the more likely there is that our methylation patterns will be lost. There will be a change. And cells in your skin, in your brain, in your heart, in your liver, in your kidneys, throughout your body will start misbehaving, which is one of the reasons tissues don't function as well as you get older. It's also one of the reasons that you die, that organs fail ultimately. So, for example, it can happen the other way around too, by the way. So you can have a normal cell with certain genes that are active. And those cells, just due to respiration, due to age, etc., certain genes within those cells can lose their methyl group. So you can end up with hypermethylation. So genes being methylated that shouldn't be methylated, genes being switched off that should be on. That can result in cancer too. Here is kind of just a very very small snapshot of various enzymes that are involved in regulating gene expression, methylation, acetylation, the rest of it. We have the DNMTs, DNA methyltransferases, I told you about earlier, that place methyl onto genes, onto cytosines. We have the HDACs, histone deacetylases, which remove acetyl groups from histones, and we have lots of other enzymes too. There are many enzymes involved in modifying the DNA and the histones to change the charge, switch genes off and on, add phosphate, for example, remove phosphate. You can switch genes on and off, their activity in many ways. There's a lot of enzymes involved, and here's just some of them. So, for example, histone acetylation will cause chromatin relaxation, unfurling of the chromatin, opening up DNA slightly, allowing various enzymes to basically bind to the DNA, and therefore switching the genes on, making messenger RNA and ultimately protein. So epigenetics and aging. This is me, by the way. I've aged slightly since then. This is 11 years ago. I have two identical twins in my arms. They belong to a friend of mine. They had just been born at the time, both premature. These are identical twins, meaning that they have identical DNA. They came from the same fertilized egg, which is split in two. But they're not identical. They're not identical. One is slightly taller than the other. Their behavior is slightly different. They're both girls. One is very tomboyish, likes football. The other one is kind of a little princess. She likes ballet. She likes pink, etc., etc. They're very different in their personalities, and they look slightly different. Very similar in terms of looks, but slightly different. And genetically, they're identical. So what accounts for these differences? Well, it's epigenetics that accounts for these differences. It's that epigenome that is different. So why is it different? Well, because they have been exposed to a different environment from pretty much the moment the egg split into two. You see, two identical twins are not in an identical part of the womb. They can't be in the same spot. One is higher than the other, lower than the other, left or right. Therefore, they're exposed to different concentrations of chemicals and hormones right from that very early stage. And these different concentrations of chemicals and hormones can cause different methylation patterns, acetylation patterns, can cause differential expression of genes. And that starts right from the beginning. And once these children are born, for the rest of their life, they will be exposed to slightly different environments, different stressors, different events will occur. They will be infected by different diseases, different viruses, etc. And this all affects the gene expression. So as they get older, they will gradually become less and less identical due to environment. Methylation patterns. Here is a young cell, here is an old cell. So in a young cell, you have usually the correct methylation pattern. The genes that should be on in that skin cell are on, the genes that should be off are off. Same with neurons, cardiac cells, etc. As you get older, due to exposure to various chemicals, various toxins, respiration, wear and tear, these patterns change. And cells become less efficient at doing what they're supposed to do, causing many of the symptoms of aging. Can we do something about this? Well, many people are trying. You might want to listen to my lecture on aging to know a bit more about that. There are attempts to basically reset cells almost to factory settings so that you can get the methylation patterns, the acetylation patterns exactly right in a cell to rejuvenate a cell. There are certain enzymes that we have found that can do this. These enzymes are necessary if you're going to make an egg or a sperm cell. You don't want it to be an old egg or sperm cell. You want it to be all young and vibrant with the correct methylation and acetylation patterns once again, so that when a baby is born, it isn't born old, it's born young. So we know there are enzymes that can help reset cells, and we know what these enzymes are. I'm not going to talk about those enzymes right now, that's for another lecture. But we, certain scientists, for example, David Sinclair from Harvard, has identified, amongst others, I should say, there's many other scientists involved, he's worked with with switching on these enzymes in certain types of cell to help rejuvenate cells with some success. So perhaps in the future, we'll be able to sort of reset ourselves to a younger pattern of gene expression and therefore rejuvenate individuals. But that's for the future, perhaps the relatively near future, but time will tell. Okay, so that's where I'll finish for now. Thank you very much for watching. You can follow me on Twitter. My hashtag is, sorry, my Twitter handle is @MarkOfTheD, MarkOfthe, capital D. Thank you very much for watching, and I look forward to speaking with you all again very soon. Goodbye for now.